Hello everyone. Today we will discuss about carcinogenesis. Now going to the carcinogenesis, it can be due to chemicals, it can be due to radiation, it can be due to microbes. In this video we will discuss about microbial carcinogenesis. Microbial carcinogenesis can be due to RNA viruses, due to DNA viruses or due to any bacteria. Now we will study about oncogenic RNA viruses. So beginning with that, first is the HTLV1 virus, that is human T cell leukemia virus. It causes adult T cell leukemia as by this name. Now it is very similar to a virus, that is HIV virus, as it has tropism for CD4 T cells, and these cells are the major target for its neoplastic transformation. Also, the route of transfer is sexual or breastfeeding or by blood products is same as that of the HIV virus. Now, the HTLV1 genome, it contains genes like GAG, pole envelope gene, LTR regions. They are very similar to other retroviruses, but in there is one more gene which is responsible for its oncogenic behavior, that is TEX gene. Now, what does the TEX gene do? So the TEX gene, what does it do is, firstly, it causes increased cell survival because it interacts with P13K and stimulates AKT pathway. This pathway leads to increased cellular proliferation. Also, it inhibits or inactivates various cell cycle inhibitors and enhances cyclin activation. Therefore, it increases cellular proliferation. And also, last is it causes genome to be unstable. It causes genomic instability, which will further lead to malignant transformation. Now going to the oncogenic DNA viruses. These are HPV virus, EBV virus, hepatitis B virus, and human herpes virus 8. Now firstly we will discuss a virus, human papilloma virus, which is largely responsible for the carcinoma cervix. But there are many types of it. The types like 1, 2, 4, 7 and 11, these cause only warts. They cause only benign warts, but there are few high risk HPVs. These 1, 2, 4, 7 and 11 are known as low risk HPVs. The high risk HPVs are 16 and 18, and they are responsible for the carcinoma of the cervix and the anogenital region. Now, what is the difference in the warts? In the warts, the HPV genome will remain in the non-integrated form from the host genome. It will be in the episomal form, whereas in cancers the HPV genome will be integrated into host genome, causing the malignant transformation. Now, studying about the various genes in the HPV which will lead to cancer, these are, there is E6 and there is E7. Basically, what will they do? They will inactivate the tumor suppressors, they will activate the cyclins, they will inhibit the apoptosis and combat the senescence. They do this by inhibiting P53 gene, which is a tumor suppressor, P21 gene, and retinoblastoma gene. These three are tumor suppressor genes. They will inhibit them and inhibit their normal function. They will not lead to apoptosis, they will not cause any growth arrest during cell cycle, leading to mutations. Okay, now going to another virus, that is Epstein-Barr virus. Epstein-Barr virus is responsible for many tumors like Burkitt lymphoma, very well known for it, B-cell lymphomas in immunosuppressed individuals, Hodgkin lymphoma, a very important nasopharyngeal carcinoma, and also some rare forms of other lymphomas. Now their role in different cancer is different. Like in the Burkitt, its pathogenesis is different. It is not directly oncogenic. You will understand by this. When the infection takes place with EBV, there is polyclonal B cell expansion. okay, to combat with this infection. What happens is, normally after it has eradicated EBV, it had to be killed by cytotoxic T cell and the B cell growth will be controlled. But sometimes when polyclonal B cell expansion takes place, mutation takes place, which is H214 translocation, very characteristics of Burkitt lymphoma, leading to this lymphoma. Okay, the nasopharyngeal carcinoma caused by EBV is not here. The role is directly oncogenic because the genes responsible are LMP1, EBNA2, and VIL1. These in this case it's directly oncogenic. Now going to hepatitis B and C virus responsible for the hepatocellular carcinoma. What happens in this is, uh, HBV, they cause chronic viral infection, they cause chronic hepatitis, okay, which lead to lots and lots of hepatocyte death. And after the hepatocyte death, what will happen is there will be compensatory proliferation of various hepatocytes, and when this regenerative process is going on due to various growth factors, cytokines and chemokines which are produced by cells, what happens is these cells not only produce growth factors, they also produce reactive oxygen species which are genotoxic and mutagenic. Due to this, mainly the tumor develops. But also, not only this, the HPV genome, now we have found that it contains a gene known as HBX, which is directly responsible, and also in HCV core protein has direct effect in the tumorigenesis. Now going to one bacteria which is known as Helicobacter pylori. Now Helicobacter pylori, this is the first bacteria to be classified as a carcinogen, and it is implicated in two cancers. Here also the pathogenesis in each tumor is different. One is the gastric adenocarcinomas and other is gastric lymphomas. Earlier, H. pylori was very known for peptic ulcer causing chronic gastritis. But what happens is, like in hepatitis B and C virus, we understood when chronic gastritis is there, it will lead to gastric atrophy. After gastric atrophy, what happens in hepatocyte regeneration takes place. But here what happens, there is a sequence, there is metaplasia, which will further cause dysplasia and then cancer will form. Okay, this is due to various immune inflammatory cells which are present, which will release reactive oxygen species. This was the one reason for the adenocarcinoma. Another reason is the one gene present in the H. pylori, that is CAG-A gene. This gene also penetrates into the gastric epithelial cell where it mimics like growth factor stimulation. It mimics unregulated growth factor stimulation. Now going to the lymphoma, we studied it also causes lymphoma, gastric lymphoma, and here the name is maltoma. Maltoma is a mucosa associated lymphoid tumor. Okay, H. pylori, how does it causes is, when the H. pylori infection takes place, the T cells, the reactive T cell, they cause B cells to proliferate. Okay, and they keep on proliferating B cells, and because the T cells, they produce a factor known as NF-kappa B, the B cells will proliferate. But when we eradicate the H. pylori, in this case, when we eradicate H. pylori, what happens is it will cure the lymphoma. It will lead to no lymphoma because the antigenic stimulus for the T cell will be removed. Therefore, T cell will not produce any NF-kappa B. Okay, and when they will not produce NF-kappa B, there will be no B cell stimulation. Okay, but after later stages, what happens is other mutations like 11-18 translocation takes place, and now even after we remove the H. pylori, the maltoma will continue to grow. Okay, okay, this was all about microbial carcinogenesis. We discussed mostly all the microbial causes of carcinogenesis. Do like, share and subscribe to my channel. Thanks for watching this video.